Thank you so much, madam. Assalamu alaikum and hello to everyone. This is the first lecture on Introduction to Computational Genomics and Bioinformatics. That is a 1 month long course. I'm thankful to Siren Network and Comstek and ICCBS and University of Sargodha for organizing this activity, this course. I will introduce myself. I'm Sayyad Bukhari. My research field is in agricultural biotechnology and microbial genomics. With that, I will start. So the course is divided into two parts. Half of the in first 8 lectures, these will be based on Linux. They will be Linux focused, but you don't need to have expertise in Linux. We will start from scratch. It will include a brief introduction to molecular biology and genomics. We will, in the second lecture, we will get started with the command line tools and how to interact with the NCBI and download large-scale data from NCBI at the command line. We will do some variant calling and genome assembly, after nucleotide identity and genome annotation. Some pangenomics, these are very hot topics nowadays, and how we can do blast locally at your own system. And we will also perform multiple sequence alignment and phylogenetic tree. All these topics will be Linux focused, and later on we will move towards R focused domain of R course. This will include introduction to R, statistics in R, data visualization, and RNA-seq data analysis. We will download in the first part of the first half of the course data that we will use in RNA-seq data analysis. Later on, we will do some data visualization of phylogenetic tree using ggtree, although there are some other tools as well. But in this course, we are focused on programming and R and Linux. So we will also do some basic machine learning stuff as well. And this will also include final exam. This is one graded R course, and participants will get a certificate if they complete some criteria, passing criteria, a minimum of 50% marks, and you have to obey the ICCBS attendance rules as well. So let's start with the differentiation of bioinformatics and computational genomics. Computational genomics refers to the use of computational and statistical tools to decipher biology from genomic sequences and later data. It also includes DNA and RNA sequence, as well as some other post genomic data like protein sequencing as well. Protein information. So bioinformatics is a collection, storage, and analysis of biological information that is particularly associated with DNA, RNA, and protein sequence. So computational genomics is a part of bioinformatics that deals just with the analysis of genetic data, that is DNA, RNA, and protein, while bioinformatics also includes some methods of collection and storage as well. So in this regard, central dogma is very important because if we want to decipher biology of a cell from its genomic sequence, central dogma is very important because the flow of information is from DNA to RNA and then to protein. What is encoded in the DNA is transcribed in the form of RNA, and that RNA gives rise to a protein, and protein gives rise to a phenotype that we can see. Traits of a cell or an individual. So you have heard about central dogma of molecular biology, that is DNA is transcribed into RNA and RNA is transcribed into translated into a protein. So there is a newer concept, central dogma of genomics, that is genomics. All the DNA information of a cell is known as genomics, that is transcribed into not all the DNA, but around 5% of the DNA is transcribed to RNA, that is known as transcriptomics. All the RNA in a cell is known as transcriptomics. So between these two layers, genomics and transcriptomics, there is something known as epigenomics. So epigenomics, these are the marks, these are some methylation or acetylation, some marks on the DNA that basically regulate the transcription of genes, and they play a major role in different diseases and development as well. So these are some mature systems that are commonly used in research. Then there are some less mature proteomics, and they are developing, and we are getting better and better in proteomics and metabolomics as well. And proteomics and metabolomics is what gives rise to a phenotype that we see, that we can detect. So what is genomics? The complete genetic information of a cell is known as a genome, and the study of the genome is known as genomics. So if we talk about a human cell have two different types of DNA, nuclear DNA and mitochondrial DNA. And DNA is made up of adenine, guanine, cytosine, and thymine. These are building blocks of the cell. So a cell has a nucleus that encapsulates a whole DNA in the form of a chromosome, and that chromosome has dispersed some parts of the DNA that gives rise to a protein is known as a gene. So why we actually study genomics? First of all, we want to know the genetic information of an organism. We wanted to know in Human Genome Project, we wanted to know the complete genomic information of humans. And we did that. We have established the genome of the human. But in addition to genomic information, we are also interested in what are the differences in the genetic makeup of different individuals. As we already talked about, and that a phenotype is a result of a genome information, because genome information is transcribed and that is translated, and the translated proteins gives rise to a phenotype. So we can see a diversity of phenotypes in terms of color, in terms of height, in terms of weight, in terms of intelligence, all the traits that that we can talk about. So what are the differences, and if these differences are actually are responsible for these differences? So we can see that any two individuals, they are 99.5% similar. They have their genomic information is 99.5% similar. They are just 0.5% different. Two randomly people picked from you people would be just 0.5% different from each other. Yet you people are astonishingly different from each other in terms of height, in terms of color, in terms of behavior, everything. So actually these 5% differences accounts around 16 million differences. These are huge differences. They are in the form of copy number variations, single nucleotide polymorphism mutations, and these mutations they accumulate and gives rise to a response. They basically influence the gene expression system, and differential gene expression gives rise to a different phenotype. Similarly, on a cumulative scale, if we talk about all the differences that may have that are present in human race, they are more than 100 million that are present in the human race. Similarly, if we talk about genetic similarities between chimpanzee and human, they are 99% same. So again, majority of the differences are at the small scale. They are single nucleotide polymorphisms and copy number variations. So these are the differences. Basically, genetic information is the same in all of the organisms, except that some of the places, some will have A and some will have a T. And these differences actually, if they are present in the coding sequence, they may have a larger impact. And even if they are not present in the coding sequence, they may influence gene regulation, and that gives rise to a different phenotype. So, what are the genetic variations? Genetic variations are of different types. A point mutation can be synonymous or silent mutation if it results into the same amino acid. If there is a mutation and it gives rise to the same amino acid, this is a silent mutation. And if it results into a stop codon and premature stop codon, it is known as a nonsense mutation. There are two different types of mutations that are non-synonymous. Missense and missense can be conservative and non-conservative. Conservative if the amino acid that is being coded after the mutation has similar physicochemical properties. This is conservative. These mutations will have less impact as compared to non-conservative mutations, because in case of non-conservative mutations, amino acid that is being encoded is not physicochemically similar to the one that was encoded before the mutation. So they have these types of mutations have larger impacts and can generate some diseases as well. So these point mutations, if they are present in 1%, more than 1% of the population, they are known as single nucleotide polymorphisms. In addition to point mutations, there are indels as well. These are larger addition or deletion of nucleotides in the genome, and there can be some structural variations as well. So selection and if there happens some mutation, it has different fates. It can be beneficial, and if it is beneficial, it will sweep into the population and become will become more populated in the population. And if it is deleterious, it will results into a negative selection, and it will sweep out of the population. If there is a neutral mutation, it can increase in frequency, it can decrease in frequency. So because it's a neutral mutation, it does not impact the genotype, it does not impact the trait, and it does not actually affect the selection system. So as I have already told that only 2% of the DNA is coded in the form of proteins. DNA is in the form of a chromosome, and that chromosome has a DNA, and there are dispersed, some sections of the DNA gives rise to proteins, and these sections are known as genes. These genes have exons and introns, and only 2% of the DNA, total DNA in case of most of the eukaryotes, gives rise to proteins. And the other 98% is non-coding RNAs. They can be ribosomal RNA, small nuclear RNA, or transfer RNA, transposable elements, intergenic regions, and introns. So what is next to the genomics is transcriptomics. Transcriptomics is the study of total RNA of our cell. So if we are interested, what is causing a disease? What are the differences in the differential expression in disease samples as compared to the control samples? We need to study transcriptomics. Although that differences are from genomics, and we can also study at the genetic level, at the genomic level, that is known as GWAS, genome-wide association studies. We take samples of disease samples and control samples, and we compare if there are some mutations that are common among disease samples and are not present in control samples. And this is how we study GWAS. But there is another aspect of this study, that is transcriptomics, that we are interested in what are the genes that are being influenced because of some mutations or because of some other reasons. And for that, we take disease samples, and we also take control samples, and we isolate total RNA from that. we fragment that DNA, that RNA, in fact, and we generate, first of all, we generate cDNA and then fragment it and attach some linkers, adapters that are necessary for sequencing purpose. We sequence it, and after sequencing, we perform some bioinformatics analysis that we will talk about in this course. So we can generate, we can find genes that are upregulated or downregulated in disease samples as compared to the control samples. So these are some glimpses of my research. So structure of a gene, you people know, a gene have many parts. It has exons, and exons are intercepted by introns. These introns are removed from the mature RNA, and the final mRNA that gives rise to a protein that is translated into a protein does not contain introns. And if it contains intron, it may result into some of the disease. So what are some major sequencing methods? One of the oldest one is the Sanger sequencing, although there was a Maxson-Gilbert method that had many issues, but the one that is very common and is oldest is Sanger sequencing. And we have next generation sequencing methods, and nowadays we are moving into an era of long read sequencing systems like the PacBio and Nanopore. We will talk about these sequencing methods as well. So if we talk about Sanger sequencing, you people know that DNA is composed of nucleotides, and nucleotides are actually, common nucleotides are deoxynucleotides, in which 3' head has a hydroxyl group. But in Sanger sequencing, we use dideoxynucleotides that does not has a hydroxyl group at 3' end. So because addition of a new nucleotide takes place at that hydroxyl group, and if that is not present, new nucleotide cannot be added. So this is the philosophy behind Sanger sequencing. What we do, we take a vial and we incorporate primers, we incorporate polymerase, we also incorporate DNTPs, all the DNTPs, and we also incorporate DDNTPs. Some are normal NTPs, deoxynucleotides, and some are dideoxynucleotides. And every dideoxynucleotide is labeled differently. What actually happens? There is a random chance that we have a primer, and we have a random chance that a new nucleotide will be incorporated at a complementary sequence of the template. And there is a random chance that normal nucleotide can be added, or a dideoxynucleotide can be added. If a normal nucleotide gets added, there is a chance that at the next step, dideoxynucleotide can be added. In this way, fragments of different sizes gets generated, and all these fragments of different sizes give rise to a different color depending upon the type of dideoxynucleotide that has been incorporated. And we run it on the gel, and we can detect it by using laser technology. What was nucleotide that was added, and this way we can perform Sanger sequencing. But nowadays we are moving towards, we have advanced systems like next generation sequencing systems, because and we are as we are moving, we are witnessing decrease in the cost of genomics and sequencing. And with that decrease in the cost of sequencing, we are witnessing increase in data generated by sequencing systems, and we are incorporating huge amount of data in petabytes and terabytes. So a next generation system, the common workflow is we first prepare, we perform library preparation. That is, we take a DNA and fragment it, we perform size selection and attach some adapters. And these these prepared library is used for the sequencing purpose, and later on we analyze the data. So there are different You must have heard about sequencing methods, sequencing by synthesis, sequencing by ligation, and real-time sequencing, single molecule real-time sequencing. So sequencing by, what is sequencing by synthesis? It is determination of the sequence of DNA by detecting the incorporation of a nucleotide by a DNA polymerase. We have a template, we incorporate new nucleotides, and as we are incorporating new nucleotides, we try to detect what nucleotide is being incorporated. This is how we find the sequence information of the DNA. And if we do by this method, this is known as sequencing by synthesis. We are synthesizing the DNA, and while we are synthesizing the DNA, we are determining its sequence. So some common methods are pyrosequencing, that is 454 sequencing, that is not used commonly nowadays. Ion semiconductor sequencing, it's not common. Illumina, it's very common, and majority of the sequencing are Illumina sequencing. And we also have PacBio sequencing. It's one of the type of long read sequencing. And in this sequencing method, sequencing is done by synthesis. So it's a long read sequencing. We can generate long reads as compared to the Illumina, which is short read synthesis system. And we also have sequencing by ligation and nanopore sequencing. We will also talk about nanopore sequencing in this lecture. So what is pyrosequencing? In pyrosequencing, we have different runs of adding nucleotides. We use labeled nucleotides, and we only use one type of nucleotide at a time. We add one type of nucleotide, that is labeled nucleotide. If it gets incorporated, it gives a signal. How it gives a signal? You people know, as a nucleotide is incorporated, mononucleotide is incorporated, and pyrophosphate is released. That pyrophosphate is converted into ATP by sulfurylase, and that ATP gives rise to luciferin with the help of a luciferase enzyme that emits a light. That light is emitted. If we add ATP and no light is emitted, for example, if we incorporate ATP and no light is emitted, it means there is no T on the template stand. We degrade all the nucleotides with the help of A pyrase. Then we will add TTP. If no light emits, it means there is no A on the template stand. We degrade all the TTPs with the help of A pyrase. And if we add GTP and light is emitted, it means at the complementary stand it was C. So we get an idea about the first nucleotide in this stand. Similarly, we use runs of different nucleotides, and one by one we become aware of the sequence of the DNA. So ion torrent is very similar to pyrosequencing. The difference is in pyrosequencing we use pyrophosphate, and that pyrophosphate is used for the detection purpose. But in ion torrent, during incorporation of the nucleotide, not only pyrophosphate but hydrogen ion is also released. In that system as well, we use different runs of nucleotides. We, for example, use ATP, and if hydrogen is released, that is detected, it means at the template stand was T. And similarly, if no hydrogen is released, it is not detected, and it means that that nucleotide complementary to that nucleotide was not present in the template system. A very common method is Illumina. In Illumina, we prepare a library, and we have primers that are attached to a cell, and we fragment the DNA and attach primers to that fragment, and these primers are actually complementary to the probes that are already attached to the cell. and they get attached and they are amplified. This is known as clonal amplification, and clusters of a single sequence, a single fragment are produced, and these after they become a cluster, they are sequenced by a method, and that method is we have fluorescently labeled nucleotides. And similar to Sanger sequencing, we have all the 4 nucleotides are labeled differently. And all the nucleotides, when get incorporated, they emit a different color of light. And we can detect that light, and we come to know that what was the sequence at that position. PacBio, a very recent development in bioinformatics is PacBio sequencing. We can generate a very long read up to 64 maximum limit is 64 kb. We commonly can generate 10 kb length of read, as compared to the Illumina that is just a 300 to 500 long read. So what we do, we prepare a fragment of around 10 kb, and we ligate adapters, and these adapters they are circular, they make the molecule circular. And we have some nano wells where we attach these circular molecules, and in these nanomolecules, polymerase is also attached to that nanowell, and later on the sequencing processes by synthesis, and it is very similar to Illumina. We have labeled nucleotides. Each nucleotide is labeled differently, and if one of the nucleotide is incorporated, it emits a light a different wavelength, and that wavelength is detected, and based on the wavelength of the light, we determine what is actually the sequence of the DNA. So the last one is this is a nanopore. This is not single sequencing. This is nanopore. In nanopore, we actually have a protein that has a very small pore, and we push the DNA or long DNA through that pore, and we have a potential difference, electric potential difference across the membrane in which the protein is incorporated. We have a membrane, and in these membrane, across these membrane, we have a potential difference. We incorporate a protein that is known as nanopore in that membrane, and we try to push that a DNA through that pore. And when that DNA is passing through the nanopore, because of different nucleotides, A, T, G, and C, they have a different structure. They disrupt the flow of the ions, and based on this disruption, we can determine which was the nucleotide, because different nucleotides will have different types of disruption in the flow of the electric current, and based on that system, we can actually determine the sequence. And these are some of the recent developments in sequencing technologies. Nanopore sequencing have MinION, a small mobile, a small device just of the size of a mobile phone that you can put on your desk, or you can put it in your pocket. You can take it into a field, and you can sequence the whole genome using that small device. So the major part of this course will be analysis of sequencing data. And as I discussed, this will be focused on Linux and some R. So I will request all of people that you install, you should have a Linux system. If you don't have a Linux system, all of you would have a Windows system. Do you have, all of you have a Windows system, right? Okay, so what you can do, you can install Linux system into your Windows system just as a software. This is as simple as you are running Mega. You must have used Mega for phylogenetic tree. So this is just like a software. What is the procedure to install that? And I want you people to install that today, so that if you come tomorrow, we can have some hands-on on the Linux, so getting started on the command line. So what you need to do, you need to go to the control panel, and you will write Windows features in your search box or the control panel. You will write turn Windows features, and that thing will appear, turn Windows feature on or off. You will click that, and there is an option, Windows subsystem for Linux. This will be unchecked. You need to check that Windows subsystem for Linux and click OK. You will do that. The system will prompt for restart. You have to restart the system. After restarting, you have to go to the Microsoft Store. This is very similar to the mobile app store. You download different software. So Microsoft Store, you will write Ubuntu or your favorite Linux environment. There are different environments, but Ubuntu is preferred. You will write Ubuntu. Ubuntu will appear. Click that, and there will be an option of get. Click on the get. It will start downloading. Once it gets downloaded, you should have a good internet system, internet supply. It will turn into a launch option. Click on the launch, and that will be launched in your system. The first thing you need to do is tell him a username and a password, but keep it in mind that username should be small, and a password, you should not forget that. Otherwise, you have to do all of the things again and again, because that username, you will require that username again and again when you are doing some stuff like installing some programs for later analysis. So don't forget the password. Keep the username small. It will help. So regarding R, I will request that you also install R. How you can install R? Just go to the CRAN, CRAN. This is a website, and you first need to download R. There will be an option of base. You need to click that base. R will download it into your system, and just like you install other softwares like ITM and VLC and other softwares, you need to click on that and install that R. After installing R, you need to go to the R Studio, write R Studio on Google, and download R Studio for desktop. For desktop, it is free, but it has also a commercial version. But you people need to install the free version. It has all the capabilities, almost all the capabilities. Download it and install it. And I wish that you install it today, because it may take time, so that you people have a system where Linux is installed, where R is installed, so that we can do some hands-on. So I think this is how it will appear when you install a Linux system on your Windows system. And this is how it will looks like if you install RStudio. It will have a different portions, console and terminal, workspace, where your variables will be present. We will talk about that, and different libraries and plots you can see. So that's all for today. If you people have any question, comment, concern. Anyone from online? Yes.
You have to install R from the App Store, like the Windows Store, or
No. From Microsoft Store, you have to install Linux. For R, you need to go to Google. You will write CRAN and click on base. It will be blue option base. Click on that. R will get downloaded, and install it just like you install IDM or VLC or other software. Similarly, for RStudio, you need to go to the Google, write RStudio, and download RStudio for desktop. And after it gets downloaded, just install it just like you installed R. But before installing RStudio, you have to install R. First is R, then RStudio. All other options will default options. Just click next, 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 just like you install a software. So, any other question? Yes. Yes, I will talk to Dr. Nadeem. I think he is communicating with you people. I can give these slides to Dr. Nadeem and he will share that. All right, anything else? So I think there is no more question. Let me see from online. There are some hand raises.
Assalamualaikum, sir.
Walaikum assalam.
Sir, I have a question regarding R software. सर आपने कहा है कि गूगल पर हमें क्या राइट करना है और आर डेस्कटॉप को डाउनलोड आर स्टूडियो को डाउनलोड करने से पहले आर डाउनलोड करने के लिए
आपने लिखना है CRAN, CRAN. CRAN लिखना है, एंटर करना है. Let me show you if it yes, I can show. You need to write CRAN. I think you people can see. Yes. So this is comprehensive R archive network. Click on that. Download R for Windows. And this is base. Just click on that. It will start downloading. Yes, download R. This is a recent version. Just click on that. It will start downloading in your system. After it gets downloaded, just click on that and install in your system. For RStudio.
Sir, one more question.
Yes.
Sir, you talk about Illumina and Solexa. Kindly elaborate it clearly. I can't understand Illumina.
Illumina. Let me show RStudio and then I turn back to Illumina. Okay, just write RStudio. Recently they changed their name to Posit, just like Twitter changed to X, so they changed to Posit. Download RStudio. Again, download RStudio. Download and install R, not that one. Download RStudio desktop for Windows, and it will start downloading into your system. First install R, and then you have to install RStudio. Regarding Illumina, let me show that. So first Illumina. So in Illumina, what we do, we have DNA, we fragment that DNA and we attach primer, we attach actually adapters. These are two different adapters. And similar adapters are also present in a cell, in a flow cell. And we merge that fragment onto that cell. And because these adapters have similarity to the probes that are already attached to the cell, they get complementary, and this is how it looks like. It gets complementary, and because this probe that is already present on the cell, it acts as a primer, and this acts as a template, and it starts incorporation of new nucleotides. After first round of amplification, actually adapter has complementary to the other probes that are already attached. It comes into a shape of a bridge and attached there, and that probe actually acts as a primer at this time, and a second round of synthesis starts. This is how this is a cycle that takes place hundreds of times, and a cluster like that appears onto the cell, and later on sequencing takes place. For sequencing, we have nucleotides that are labeled differently. All the 4 nucleotides are labeled differently. Whatever nucleotide is incorporated, it gives a light of a different color, and that light is detected by a detector. And this is how we perform sequencing by Illumina. Is it clear?
Yes, sir. Thank you so much.
So I think Abdurrauf has a question. I think he's my class fellow. Let's see what he has to say.
Hello, sir. I'm not your class fellow. Can you hear me?
Yes, I can.
Sir, I have some questions. First of all, the first question is why DNA replication starts from 5' end, not from 3' end?
Because next nucleotide gets incorporated on a hydroxyl group, and polymerase also has a tendency to move DNA from 5' to 3' direction, because the newly coming nucleotide gets incorporated on 3', and it has to attack on the pyrophosphate, on the phosphate, triphosphate. If it starts from 5' end, what will happen that triphosphate will not be present. Actually, triphosphate is necessary, that comes with the incoming nucleotide. If it starts to attach DNA, new nucleotide at 5' end, there is no triphosphate at that end. That is the reason it is not possible.
Okay.
So
The next question is
yes.
okay. I understand. The next question of mine. I have multiple some questions. Capping of protein or messenger RNA. Can you explain this? Because I am confused about this. These days I am reading the genetics.
Capping of messenger RNA is one of the post-transcriptional changes. There are many post-transcriptional changes like splicing, capping, and sometimes RNA editing as well. I don't think I should go into the details of the molecular mechanism of capping, but the idea is in eukaryotes, 7-methyl guanosine, I suppose, gets attached to the 5' end, and it has a major role in regulation of gene expression. It basically controls the gene expression. So I think
Okay.
okay. So nanopore, regarding nanopore, we have a membrane. In that membrane, we have a protein that is known as nanopore protein. It has a small pore, and only DNA can flow through that pore. And we also have a electric potential difference across the membrane, and because electric potential means ions can move across the membrane, and if this DNA and these ions actually move uninterrupted if there is no DNA. If DNA starts passing through that pore, the supply of ions from one side to another side is disturbed, and because different nucleotides disturb that flow of ions differently, and based on that difference we can determine what is the nucleotide that is flowing through that nanopore. Is it clear now? Thank you. Okay, so anything else from online?
Yes. Can I have one more question? Sir, kindly describe the frame shift. What is frame shift?
So I think these are not in the scope of the course, but frame shift is if we have a mutation, and because of that mutation should be deletion or addition. If there is a deletion or addition, the triplet codons gets disturbed, And because of that triplet codons disturbance, a protein of different sequence gets translated, and that is completely different protein, and that protein actually will have a different impact. It can cause a disease, it can have a different impact. Sometimes it also results into premature stop codon. So in that case, a truncated protein will be produced, and that will be most of the time non-functional.
Thank you so much.
Okay, let's take 1-2 more questions from online. So I unmute HP. I also unmute Akmal Zubair. So Akmal Zubair or HP, if people have a question.
अस्सलाम वालेकुम सर।
वालेकुम सलाम।
सर ये लिनक्स सॉफ्टवेयर के बारे में दोबारा समझाएं कि इसको इंस्टॉल कैसे करना है। और दूसरी बात सर, ये जो पार्टिसिपेटेंट है उनको सर काइंडली रिक्वेस्ट यही है कि आप लोग कोई जो है ना टेक्निकल क्वेश्चन कर लिया करें। इस जो क्वेश्चन अभी कर रहे थे कि फ्रेम शिफ्ट वगैरह ये तो सारे गूगल पे मिल जाते हैं, जस्ट लिख लो, वहाँ पे सब कुछ जो है ना 2 लाइन में मिल जाता है। इससे वैसे टाइम वेस्ट हो जाता है।
Don't worry. If I know an answer, I can answer that. If I don't know, I can say sorry. There is no problem in that. Allah knows everything. I know many things, but not everything. So I can say no when I don't know. So no issue. So your question was to show how to install Linux. So
Yes, sir.
I show Linux. Let me see if you people can see. Yes, you people can see. It's Windows 11. This is control panel. You need to write Windows feature on or off. Windows features on or off. That will pop up. In that, you need to click on Windows subsystem for Linux. If you do that, let's do that. It will take some time. It will prompt you for restart. If it does not prompt you for restart, you have to restart. After doing that, you have to restart. It is prompting for restart. I will not install Linux, so I will not restart that. After that, you have to go to what is Microsoft Store. Microsoft Store. In that store, you need to write Linux or Ubuntu, maybe directly Ubuntu 1.20. There will be different options. Just click on any of this. Just let's see, click on there. Click on get. If you click on get, after some time it gets downloaded, and you have to click. It will turn into launch. Launch it, and window will window just like that will launch, and it will prompt you for username and password. You have to provide username and password, but don't forget username and password. That is how you have to install Linux. So I suppose everything is clear, and we are also short of time. So thank you so much, everyone. See you tomorrow. I think tomorrow it will be 10 a.m. There will be some issue at 10 a.m. today. So tomorrow we will meet at 10 a.m. Thank you so much. Good afternoon.